Cassel, and today I will be discussing my gut microbiome project. So, gut microbiome is a collection of microorganisms found within the animal gut. In general, microbiomes can be found within all of the human body, and they consist of a combination of microbiomes: bacteria, bacteriophage, fungi, protozoa, and viruses. Within a human, there are 10 times as many microcells compared to the human cells. But microbiomes can be found all over the body, and each microbiome has a unique function. For example, the gut microbiome can help people identify illnesses within the body, such as diabetes, obesity, and even specific cancers. So, my first example of gut microbiome, Dr. John Crayon has been studying the different microbiomes within mice. He studied mice that were born in a sterile environment, which can have much different microorganisms compared to a mouse that was born in the wild. He found that the mice that had the missing microbes of a wild mouse resembled human functions such as depression, anxiety, or even autism. But once the mice were injected with a benign bacteria, some of them resumed normal behavior. This demonstrates the direct correlation between the mouse's behavior and the gut microbiome. Another example I found of gut microbiome is that the gut microbiome has been linked to obesity. Researchers at Lund University have correlated obesity with a specific gut microbiome that fluctuates the weight of a person. So obesity can be identified through a person's metabolism. An obese metabolism would have a high body mass index. They later discovered that an obese person has 4 specific bacteria in their gut microbiome: Blightia, Duria, and Romanococcus in the Lactobacillus family, as well as CHA98. Now they are focusing on the composition of gut microbiome in order to control a person's obesity or weight in general. So the question of our lab was what DNA microorganisms are present in our sample, and our hypothesis was that the 2 samples will be similar due to them both being mammals, but they may differentiate due to their diets as well as their environment that they are in general. So we also used positive and negative controls. Our positive control was the E. coli, and our negative control was diluted water. The subjects we chose were an African pygmy hedgehog and a guinea pig. We chose these based upon their similar size and as well as they're both mammals, but we also chose these because they have different physical features as well as diets. A guinea pig intakes a vegetarian diet, such as like grass, fruits and vegetables, and African pygmy hedgehog does eat a vegetarian diet, but also has insects of some kind in their diet. So the materials that were used were the Chi kit, which isolates DNA that we used in week 1, the fluorescence spectrometer to determine the concentration and purity of our sample, DNA sequencing via the Illumina MIS-K, the Zymo kit, which increases the concentration of the DNA by reducing the sample volume. We also use the UV spectrometer, which is also known as the nanodrop. It reads at 260 nanometers to calculate the DNA concentration. We also use the Amplicon PCR. It is used to amplify the regions V3 and V4 of the specified gene, 16S ribosomal. The 16S RNA gene, which removes the primers. We also use the Agross gel electrophoresis, which separates the macromolecules such as DNA, RNA, and proteins. So it would have